אוקיי, אז ערב טוב עוד פעם לכולם, ובואו נתחיל. אני רוצה עוד פעם לדבר כמה שניות על ה-assignment. גוגל שיט מתמלא בקצב מאוד איטי, ובמיוחד התאריכים הקרובים, מיד אחרי חופשת הפסח, הם ריקים לגמרי, והייתי מאוד מבקש כמה מכם יתנדבו להיות מציגים ראשונים. זה מאוד חיוני שנקבל פיזור מתאים של כולכם לאורך כל 9 שבועות שאתם מציגים. דיברנו כבר כמה פעמים על מה התוכן של המאמרים, ואתם שולחים לי מכתבים. כן? Oh, sorry. Sorry. I don't know. I kind of switched to Hebrew. My apologies. All right. So, back to the assignment, we talked about this several times. I asked that you populate the initial days. We are missing people immediately after the Passover break. So please go back to the Google Sheet and put your name down, and hopefully you choose the early dates. We need some volunteers to do that. You have been emailing me your thoughts about which papers to present, and I authorize or ask some questions. In principle, when you come up with a topic, you need to think it cannot just be a simple paper from your own research that you know very well. That will not do. We are quite a diverse group here, and you want to appeal to everybody, so you have to have a very general concept, either, problems with the scientific method or misconduct or whatever have you, or an exceptionally good paper that is of interest for everybody. So I've been communicating with you, and I authorized quite a few papers, and I also asked a few of you to maybe think about something else. So again, we are looking for a good story which will be of interest to everybody. So the Google Sheet, as I said, is populating slowly. I need you to commit to a date and a topic, and the topic has to be approved. And as I said, we don't have enough speakers for end of April, beginning of May. We need at least 3 speakers for each of these dates, so please sign up. And 2 more announcements. First, next week I will be in the East Coast in Baltimore, and I'm going to teach from there. I hope that there will be no glitches, but I'll hopefully get good internet connection, and I will work from my laptop. But basically, we'll have a regular meeting. That's for next week. And today, after posting the lecture of today on Moodle, I'm also going to post an exam. This is a take-home exam that you will submit by the end of the week, based on something that we'll present today. So please submit your QM by midnight of Sunday, April 3rd. So it will be a PDF posted online, and Leon, if you can help me set up an assignment. The assignment will be a place where you just drop your answers to the test. That should be a PDF file that you just drop in Moodle. Okay, so back to the topic. Any questions about administration, announcements, and so on before we start? The question is, what is QM? QM is a short for the word cumulative. It's really a historical term for some kind of a course we had here at UCLA, where we gave exams to students where they had to analyze papers from different fields, not their own. So you are going to experience such one paper. It will be my paper, and you will answer questions. You'll have to read and answer questions. It's outside of your comfort zone, and the idea is to see how well can you understand reading a paper not in your own field. Any other questions? Oh, no. If no more questions, let's go on with the lecture. So last time we covered both the Watson and Crick paper, and then we covered the Forrester paper, and we talked about energy transfer. And today we are going to continue with energy transfer, and that was basically a segue into my own research. We said that fluorescence resonant energy transfer, or Forrester energy transfer, is a dipole-dipole interaction on the molecular scale. It's like transmitting and receiving antennas, where we excite a molecule, and the free induction decay, if you want, of the first molecule is the radiation that is absorbed by the other dipole. It's not a real absorption event, it's just a radiation-less energy transfer. And we did not develop the theory, but we wrote down the results of Forrester. The rate for energy transfer scales like 1 over the distance to the 6th power. And basically, this is the Jablonski diagram. We excite one molecule, and that excitation moves to the other molecule, and radiative relaxation from that other molecule. So what we are going to do today, we are going to follow up on that story. First is the concept of molecular ruler. Once the theory was there, and the theory was developed at the end of the 40s, 46, 48, it took some time actually until people really started to use it in a practical fashion. And the first demonstration was in this paper. Well, in fact, this is not the first demonstration, it's the second, and we'll talk about this in a second. This is by Lubert Stryer, a very prominent biochemist from Stanford. And he's, at that time, I think, PhD student, Richard Hageland, who later on went to develop basically technology and a company called Molecular Probes. That was, at that time, the only company that made fluorescence dyes, a large selection of them. Eventually, it was purchased by Invitrogen, nowadays Sigma. But when I started my business with biophysics, the only place to get fluorescent dye was from this guy, Richard Hageland. They published in 1967 a seminal paper in PNAS. This was communicated by Arthur Kornberg, who was an Academy member. And if you know this name, this is a Nobel laureate from Stanford. And his son is also a Nobel laureate, Roger. Roger has a both position at Stanford and Hebrew U, and he actually stays quite a bit in Israel. And what they say in this paper, we report here an experimental study of the dependence of energy transfer on distance. So Forrester had this prediction that the energy transfer efficiency depends on the distance to the 6th power, 1 over the distance to the 6th power. It took basically almost 2 decades to demonstrate this experimentally. The observed efficiency of energy transfer as function of distance is in excellent agreement with the r to the minus 6 dependence proposed by Forrester. What they've done, they synthesized peptides. In fact, they made homopeptide from L-proline. This is the amino acid, and they made basically a polymer of L-proline going from 1 monomer and up to 12 monomers. And they put donor and acceptor on the ends of this polymer. Note the date, this is published May 1967, just a month before the 6-Day War. And this is the most important result in this paper. You see the famous Forrester dependence of energy transfer as function of distance. And this curve is this 1 over 1 plus r over r naught to the 6th power. In fact, if you convert this to log-log scale, you get a linear dependence. If you fit it, you find that the slope is 5.9, pretty damn close to 6 as predicted by theory. However, and I was totally unaware of this, there was a paper in February of the same year, 3 months before, from the Weizmann Institute by Mary Wilczek and his collaborators. And they've done a very, very similar experiment. This is not the full curve that you have seen, it's only the tail at larger distances. But they basically have done the exact same thing. Quenching was observed in the series of tryptophan and glycine, tyrosine with ionization of the phenolic group. The efficiency of quenching decreases with increasing molecular size. Quenching is attributed to radiation-less energy transfer between the tryptophan and ionized tyrosine residues. So this is just to show you that in science, oftentimes, names, famous names, better journals, good university, good PR can play a very, very important role in terms of who eventually gets the credit. So Stryer and Hageland are considered the ones to basically confirm fourth cell theory for the first time, although you can see here maybe not as good paper and maybe not the full range, not all the controls and so on, but the actual observation of this dependence was done before, was done actually at the Weizmann Institute. In fact, Weizmann, after this paper from Stanford, Weizmann took really a main role in developing FRET as a tool further on. In fact, our fourth president, Ephraim Katzir, was heading this group at the Weizmann, and they have done a lot of good work on FRET. A famous student of that group is Elisha Haas, who later on became a professor at the Bar-Ilan, now retired, and had done fantastic work on FRET over the years. So there is actually a very strong Israeli component to this story. Okay, so just to summarize, we put donor and acceptor close to each other. There is resonance between donor emission and acceptor absorption. And if you put them close together, you have this energy transfer. Practically what it means, you can go with cuvettes to your fluorometer, do simple spectroscopy. If your sample has short distances, the spectrum that you'll measure will be more acceptor, very little donor spectrum. If they are far apart, more donor spectrum and little acceptor. And if the distance is basically 50% energy transfer, the Forster radius, you'll get equal amounts of donor and acceptor. So basically, using a spectrometer, you can measure molecular scale distances. That was a real paradigm shift, because until then, the only way to measure short distances was basically crystallography, and you had to have a crystal, which is periodically ordered. You couldn't really measure distances in something that is not ordered. And cryo-EM was not developed at that point. It was very difficult to measure distances on macromolecules in any other way. So that was really a big deal. Ever since this curve from 1967, people started to use fluorescence resonant energy transfer to look at molecular interactions and molecular distances. And there are many formats for this. So you can basically look at association, binding, unbinding, no signal, yes signal. You can look at enzymes that cut DNA or cut peptides. This is proteases or nucleases. And you can basically use this for many, many different things. And so from 1967 until 1996, that's basically the current application where lots of biochemists are using this method, but they're using it on the ensemble level. And they're using these to measure basically distances. And now I'm going to try to show you something that I hope will work. Just a minute. Let's see if I can get this to work. I'm going to stop share and just a minute. See if I can share this. Everybody can see this? Turn your mics on and let me know if you can see this.
Yes.
Okay.
Yes.
Yes.
So this is a piece of software written by my colleague Xavier Michelet about 22 or 23 years ago in one of the very first versions of LabVIEW, a graphical software tool developed by National Instruments, which we have evolved over the years because I still like to show these simulations after so many years. It really demonstrates that jump from what Strayer and Hagelin did and what my group was able to do. What you see here, imagine that this is a test tube, a two-dimensional test tube with many pairs of donor and acceptor. They are fluctuating. The distance between donor and acceptor on each of those virtual strings is fluctuating. And we are putting this in basically a cuvette, go to the fluorometer, and we measure the spectrum in two detectors. We have, let's say, a dichroic mirror where we split the spectrum of the donor from the spectrum from acceptor, and we collect the light. These are the two curves that you see here in green and red as function of time. So basically, this is almost not changing. And if we were to calculate from those two measurements, the FRET efficiency will get basically one value. If we now reduce the number of those pairs in our test tube, suddenly the time signals here get much noisier, and we are getting distribution of FRET value. And if we go to one pair, now we really see the fluctuations in the distance, and we go to a full FRET histogram. So that's the idea. If we are able to look at single molecule FRET, we can look at dynamic changes in the distance. And this is really a paradigm shift again, because not only that we can measure static distances, we can measure changes in distances, and therefore we can transition from static structural biology to dynamic structural biology. So that's a story I'm going to tell you today. Let's share again the Any questions about the simulation? All right. In order to do that, what we have done was to use the near-field microscope. This is a development originally by Aaron Lewis, who was a professor at Cornell University, and in around 1987 made Aliyah, and he immigrated to Israel, to the Hebrew U. He developed this idea of super resolution imaging way before all the other methods that you're familiar with for super resolution imaging. The idea was to have a small aperture and have force lights to an aperture much smaller than the diffraction limit of light, and this very close to the sample. That was implemented by optical fibers. And in 1993, Eric Betzig publishes a very important paper in Science demonstrating that with this instrument, he can measure single molecules at room temperature. The belief at that point was that you need a very small volume of excitation to see single molecules, because the single molecule signal is more or less fixed, while the autofluorescence, the background, is dependent on the volume of excitation. So in order to see single molecules, you have to have a very small excitation volume. And it was believed that this can be done only by the near-field microscope. Later on, we and many others found that that's not really correct. You can go to much more larger volumes of excitation, like a conventional confocal microscope, and still get pretty good signal from single molecules. But at that time, that was the common belief. Eric Betzig, by the way, was a PhD student of Aaron Lewis, later on moved to AT&T Bell Labs, and continued to develop this technology. When I moved from Bell Labs to LBL, Lawrence Berkeley National Laboratory, I was very interested in scanning, near-field scanning microscope, and we built one. And we had one ready when Eric actually published his science paper. So it was very natural for us just to try to see if we can see single molecules the same way he did in his paper. So this is an image taken from his paper. You can see here basically a scanned image, raster scan image pixel by pixel, and you see bright spots. Those bright spots are single molecules. I really highly recommend you guys read this paper because it's cited in his Nobel citation. This was really a fantastic breakthrough to the whole field. He really is more well known for his invention of the palm or localization microscopy concept, but he started to think about this concept while he was doing his PhD with Aaron Lewis. Here with an near-field microscope, he could see single molecules, and you can see that the shape of each of those bright spots is different. And the reason is the field lines coming out of this optical fiber coated with metal have this shape. This is bottom view and this is side view. Now, if you put a dipole, that's a molecular dipole of a dimolecule on the surface, and you scan with this electrical field distribution, you scan the dipole, you are going to actually map with a molecule, you are going to map the field lines coming out of this fiber. So depending on the orientation of the dipole, you can get a nice Gaussian shaped spot, or you can get elliptical, or you can get crescents, equal or different to crescents, 4 crescents. And if you look carefully here, you can see all of these. And basically because of that, you can locate in space the orientation of the dipole of single molecules. This was such an important and paradigm shifting report because nobody believed that you can see single molecules at room temperature. The belief was that the signal is not good enough, and there are many factors that will make the measurement very, very noisy. However, he shows that there is fantastic signal to noise and signal to background here, and it's possible. Since we just finished building our own microscope, we put molecules on the surface, and we wanted to test if we can see molecules as he could. And sure enough, yes, we could. So what you see here is, I don't think we even published this result. This is a field of molecules. In fact, much more difficult molecules to detect that Eric Betzig used. Those are coumarin dyes that are excited in the blue, where you have much more autofluorescence from the glass. And we raster scan with a fiber tip, and you can see all kinds of shapes again. What is interesting here, not really the orientation of the dipoles, but the photophysics that you can see. Here you see a nice molecule that is up and running the whole time we scan. But here you see a molecule that basically bleaches, it photodegrades in the middle of the scan. Here you see a molecule that went to a dark state and came back. And here you see a very noisy molecule. These topics of photophysical fluctuations in dyes have occupied us and many others for many years ever since. And the next jump was, okay, if we can see single molecules, what we can do that Eric Betzig has not done. And we came to the conclusion that it would be interesting to look at interactions between two molecules. Initially, we were very naive and we just threw in two kinds of molecules on the surface. That did not work really well because the chances that you'll find two molecules at the right distance is very small. And we came to the conclusion that we have to put two molecules one next to each other in a controlled way. We started to read the literature. We were aware of Try and Hagoland paper using peptides, but that seemed to us at that time quite difficult. And people started to do some really interesting work with DNA. It was possible to put dye molecules on DNA. We went to Paul Selvin on the Berkeley campus to help us with synthesizing DNA labeled with two dyes. And this is what you see here. So we made basically this DNA molecule with tetramethylrhodamine and Texas red. Here you see the spectrum, absorption and emission spectrum for those two dyes. In green, you see the spectral overlap that is used to calculate the Fourier radius. And we put this now molecules on surface of glass and use the near-field microscope to scan and see if you can see energy transfer on the single molecule level. So as I said, we initially used this near-field microscope just because we and everybody else at that time believed that that can be done only with a near-field, but later on, it was about a year later already, we did confocal work, and you are now familiar with all these other methods of total internal reflection microscopy and many others, light sheet and so on. So here is our first paper. This is 1996. Let me remind you, Stryer and Hageland are 1967. So this is almost 30 years after, where we go from ensemble to single molecule. So Forster to Stryer is 20 years, and Stryer to our work is 30 years. And we are almost, we are basically doing this, and many others in the field are doing single molecule FRET for the past 25 years. So the rest of the talk today will really show what it can do and maybe a little bit about the future. Any questions so far? Okay. So this is a figure taken from this paper from 1996, where you see a spectrum from this DNA molecule labeled with the two dyes as function of time. Time equals 0 is here. So initially we see spectrum which is red, and at 3.5 minutes, this spectrum switches suddenly to green. And this, I will argue, is an evidence for single molecule FRET. At about 5 minutes, this green spectrum also disappears, and there is a little bit bump left. So this is basically autofluorescence that needs to be subtracted. This is donor-only emission, and this is acceptor-synthesized emission. Here is this minute. At 3.5 minutes, you see basically three frames. You see the red frame, then there is an intermediate frame where that transition happened in between, and then you see the green frame. And basically, all the energy that was basically going to the acceptor, since the acceptor photodegrades and is no longer, basically goes back to the donor. So that is a proof for single molecule FRET. You can also see the opposite events on some other molecule where the acceptor bleaches first and you're left with, where the donor bleaches first and you're left with direct excitation of the acceptor. And soon enough, after Eric's work, Jay Troutman, also in AT&T Bell Labs, have shown that you can actually see single molecules in confocal. And basically, for us, it was very easy. You just remove the fiber and you have a confocal configuration. This is a scanning stage confocal microscope. And same DNA molecules. Now we don't need the near field aperture. We just have a focused laser beam and we scan and we collect data on 2 different detectors, and we basically false color and overlay those two. And you can see that all these spots show both green and red. So those are FRET pairs. And you can see the same type of acceptor bleaching and donor emission goes up, as I showed you before in spectrum. So that was the beginning for us of a new era, because until then I was really an applied physicist, solid state physicist. I built this microscope to see excitons in quantum wells. But the idea that we can now do single molecule FRET and the idea that we can look at dynamic changes of distances on macromolecule was so appealing that basically I decided to become a biophysicist and started to study by myself biology and chemistry, and through interaction with many collaborators, learn how to use these tools to answer interesting questions in biology. So why acceptor photobleaching gives a proof for single molecule FRET? Any other ideas? I actually basically said why it is approved, but let me go over this again. Okay, so this is the Jablonski diagram. And as we said, we excite the donor with a blue laser. The donor can relax by radiative transition, giving a green photon, or by a non-radiative transition, by vibrations. But if we put an acceptor molecules close to the donor, this radiative transition almost doesn't happen, and we get this molecule to get excited, and this molecule can either relax by radiative transition in the red or non-radiative transition. So if the two molecules are really close to each other, we are mostly going to see red emission. But if we are killing this molecule suddenly, because it photodegrades and it doesn't absorb light anymore, this molecule is excited and it has to relax. And therefore, all the energy is going back to the donor. So this is basically what you see here. We are exciting the pair. We are seeing mostly a red emission, but when the acceptor photobleaches, the red emission goes away and all the energy comes to the donor. So that really proves that process. Okay. Later on, we were able to show that this can be done not only on the surface, where we park our tip or our confocal spot on one molecule and we collect a signal, but we can actually have molecules in solution. We have a focused confocal spot in water, and molecules come in and out of that focal spot by diffusion. So when you do this experiment, in contrast to the previous experiment, you don't get this constant signal as function of time until photobleaching, as you've seen in the previous panel, but you see bursts. Each of these bursts here represent a molecule coming in and out of the focus. You make sure that the concentration of molecules in your sample is low enough. It's about 100 picomolar. This is very low concentration. The confocal spot volume is about 1 femtoliter. It's about 1 micron by 1 micron by 1 micron. And you make sure that the concentration is such that that volume in space is occupied at very low probability with the molecule. So that this is how you come to 100 picomolar concentration. So every now and then you get a molecule. So you have a baseline and you see bursts, and each burst represents a molecule coming into the focus and going out. If you have two detectors, you can now get photons from donor and acceptor. And now each burst can be analyzed for FRET efficiency. Yes, there was a question?
Yes, we have a request from the chat to repeat the answer and to explain what is photobleaching.
Okay, so photobleaching is a chemical process where the molecule degrades. These are organic molecules. There are carbon bonds there. If you have radicals like triplet oxygen, you basically can break bonds. Once you break bonds, that dye molecule doesn't function as a dye anymore. And when the acceptor cannot function, cannot absorb light and emit light, it doesn't serve as a way to de-excite the donor anymore. And the donor has to basically relax by itself by emitting a green light. Does that answer the question? Any other questions? All right, let me continue. So in this format, we can actually look at tens of thousands of molecules in a short period of minutes compared to very, very slow throughput in the immobilized format. And what we get here, we don't get time trajectories, but we get Fred histograms that I've shown you with this simulator. We basically can look at populations of molecules. So here we made DNA where the distance between donor and acceptor is 7 bases, 12 bases, or 19 bases. When the distance is 7 bases, energy transfer efficiency is very high. So you see this very high peak at energy transfer close to 1. You see a smaller energy transfer for 12 bases. And for 19 bases, this is already a very large distance. We see even smaller peak. And we also have this peak that is basically an artifact. The fact is, when we prepare those DNA molecules with donor and acceptor, we hybridize 2 strands. And oftentimes you'll have one strand at axis compared to the other. So there are some molecules which are donor only. And I'll show you in a second how we handle this. So basically, we can measure distance on a molecular level using the single molecule Fred in solution. And we basically repeat the Stryer and Hagoland paper that you've seen from 1967, but now with DNAs and doing this on the single molecule level. In each measurement, we acquire for several minutes and we look at many tens of thousands of molecules. However, our data is not as good as Stryer and Hagoland. You see that the dependence is not really 1/r to the 6th power. And this point we'll revisit again because we did this and we were not the only one to notice this. So here is a paper by the group of Bill Eaton from the NIH. And this is early 2000s. They basically repeated the exact experiment of Stryer and Hagoland using polyproline peptides rather than DNA as we did. And look at what they say here. The observed mean Fred efficiency agree with those determined from ensemble lifetime measurements, but differ considerably from the values accepted from Forster theory with polyproline treated as a rigid rod. So one of the issues with the Stryer and Hagoland paper and our paper with DNA is that although people thought that polyproline is really a stick, a rigid rod, apparently this measurement that Eaton is showing here contradicts this. And maybe we need to think about polyproline not as a really stiff rod, but something else. So here is their experiment. Now you can see Fred histograms using our method. And they look at different number of amino acids. And they plot this graph. And again, very strong deviation from 1/r to the 6th power. They actually make a fit, and the fit fits something more like 1/1 plus r to the 4th power. So there is a discrepancy between what we measure in single molecule Fred and the Forster theory. That wasn't satisfactory enough, so they kept on going. And in a follow-up paper from 2007, they investigate the issue further. And they find that about 30% of molecules in water contain internal cis proline. So proline is a molecule that has, if you put polyprolines, you can get 2 configurations of the bond between 2 adjacent prolines, and you can get cis and trans, and there are some angle changes. Stryer and Hagoland assume that they have a stiff rod and all the prolines are aligned in the same angle. And what Eaton finds here is actually that 30% of the prolines have kinks in the connections between adjacent polyprolines. We find that the fluctuations of the chromophores attached to long flexible linkers also play an important role. So what I neglected so far to tell you is that when we put those dimolecules on polyproline or on DNA, we actually use 6 or 8 or 4 carbon linkers that are very flexible. So the dyes themselves can fluctuate quite a bit. So basically, bottom line is now we have a question mark on Stryon-Hagelin results, which were ensemble results, they were in perfect agreement with theory. But when we repeat those experiments 30 years later with much better tools, we actually see a lot of discrepancies. We can explain those discrepancies. We can simulate, we can really give explanation. The theory is rock solid, but the experiment does have issues. There are some artifacts involved with how you put the dyes, what are the linkers, are there cis-trans transitions, and so on. But one could ask himself, how come Stryon-Hagelin data is so good while Eaton data shows all the problems? So I just leave this open. I have my own thoughts, but just, an historical curiosity. How come they have such good data? Bottom line of all of this is we now have a tool that allows us to look at moving parts of molecular machines. And this is really, as I said before, a paradigm shift, because we can now think about a subfield of structural biology, which I dub as dynamic structural biology. Any questions so far? Okay. All right, this simulation doesn't work. This does work. So this is a very old slide that I used to show many years ago when we talked about single molecule thread. Here you see, this was early days of using molecular dynamic simulation to look at protein folding. The Panda group at Stanford developed something called protein at home. That was basically following this concept of SETI, looking at signals coming from space using CPUs of many, many people around the world. Panda used the same concept to basically perform very difficult molecular dynamic simulations. So here you see an alpha helix folding. For some reason, this simulation doesn't work anymore. This is a work by the George Oster group in Berkeley, where they looked at the subunit rotations of the famous F1, F0 ATPase molecule. This is basically the molecule in the membrane of mitochondria that makes ATP, the energy that we consume. And this is just a depiction of an enzyme that processes DNA. It can be a DNA polymerase, RNA polymerase, nuclease, and so on. So the idea is, if you can look at moving parts, we can look at protein folding, we can look at enzymes, and while they are doing the work, and we can basically follow the moving parts. So the next logical step, and many people jumped at this, is to really look at the most important enzymes that are responsible for life. And that brings me to the central dogma of biology. The genetic code contains all the information about genes in every cell of our body. When cells divide, there is an enzyme called DNA polymerase that basically is responsible for copying the genetic content and make a daughter cell with the exact same copy of the genetic code. When genes are supposed to be expressed and become proteins to do particular function as function of all the signals that the cell integrates and make decisions, there are two steps to making genes. The first one is transcription, done by RNA polymerase. Basically, it reads a particular open reading frame. It binds to a promoter, and it makes a copy of that genetic information, which is one protein long, into a messenger RNA. So that conversion from double-strand DNA to a single-strand messenger RNA is done by RNA polymerase. And then this messenger RNA is read by the enzyme called ribosome to make proteins. Obviously, each one of these enzymes got at least one Nobel Prize, if not several. These are really the very, very basic enzymes that are responsible for life. And later on, it was discovered, actually at the Technion, that there are some enzymes also that are responsible for degrading proteins. So this is Hershko and Chachanover in 2004. The ribosome also, Weizmann Institute, Ada Yonah. Transcription is Roger Kornberg. I already mentioned his name. And I forgot the name of the guy who got the Nobel for the DNA polymerase. I think there are several. And what my group, after developing this method of single molecule FRET, what we have concentrated on is on this enzyme, RNA polymerase. And what you see here is the transcription cycle for bacteria. In this case, we are talking about E. coli. So just for you to understand the cycle, we start with a core enzyme and a transcription factor called sigma. Sigma binds to the core. Sorry, let's start here. This is a core enzyme, and this is sigma. Sigma binds to the core to form holoenzyme. This is now an enzyme that is capable to bind promoter DNA. This enzyme then comes and binds to a start of a gene called a promoter. This is a particular sequence that denotes the start of a gene. Through some isomerization steps, it separates the double-strand DNA and opens a bubble, which is about 10 to 15 bases long. This is called the open complex. And now if you provide this enzyme its fuel, basically nucleotides, NTPs, it will start to transcribe. But initially what happens, it's not very possessive. It will start to make transcripts, lose them, try again, and it is somehow locked in a state that is not productive. It makes lots of short messenger RNA that are not good for anything. And only by chance, if it gets some excess energy, some excess thermal energy, it manages to go over the hump and go into a stable elongation. This process of abortive initiation, we'll talk more about it. And in fact, the exam you're going to take is on this process. Think about an old Beetle car going up a steep hill, engine shut down, and the car slides back. Try again, and at some point it manages to go over the top of the hill. Once it's doing that, it's going downhill, and it's going all the way to the end, to what is called the termination of transcription. So the enzyme is very unpossessive in this abortive initiation cycle, but once it goes into elongation, it's becoming very, very possessive, and it will commit all the way to termination. So as we said, we have this abortive initiation. The next step is promoter escape, and now we are processing and go all the way to elongation. Once we get to termination, sigma falls off, the messenger RNA falls off, and the components are ready to start this cycle again. So this is a bacterial transcription cycle, and our tool is this confocal single molecule FRET, as we described before. However, as I already hinted before, when we do this simple single green laser confocal spot, and we get those bursts, we are able to measure FRET histograms, distribution of distances, but we cannot distinguish large distances from donor-only molecules. We wanted to solve this, and for this, instead of doing one laser, we actually have in the confocal spot 2 different color lasers. One green laser that excites the donor molecule, and the other one is red laser that excites directly only the acceptor molecule. And we alternate between those 2 lasers very quickly on the microsecond timescale as the molecule diffuses, and that gives us extra information. So instead of having a single histogram of FRET efficiency, with this extra information from the red laser, we can actually have a 2-dimensional histogram, where one axis is the FRET efficiency E, the other axis is what we call stoichiometry, and that allows us to separate donor-only molecule, acceptor-only molecules, and donor-acceptor molecules at different distances. And we are interested only in these molecules because here we can see changes in distances. So we want to filter out these molecules that we cannot purify basically with conventional biochemistry means. In our test tube, we have all kinds of molecules, but we can now filter them electronically and just look at the molecules we are interested in. So this is called alternating laser excitation, or ALEX spectroscopy. And with these tools, we can now basically go and try to answer some questions. So here is again this two-dimensional histogram. And we use the bacterial transcription system just to demonstrate this first. So here what you see is what we call ALEX histograms. Again, FRET efficiency versus stoichiometry. The donor-only molecules were already filtered out. You see only FRET-relevant molecules in the squares and some acceptor-only molecules. And here you see the enzyme bound to the promoter. The bubble is opened, and you get some distance. So this is what we call leading edge FRET. So the front of the enzyme is labeled with donor, and downstream DNA is labeled with acceptor. And the distance is such that we get FRET efficiency of 0.3. Then we can add not all 4 nucleotides, but actually subset of 3 nucleotides. And the first 11 bases on that promoter don't contain that 4th nucleotide. Therefore, we march the enzyme up to position 11 and we stop. And you can see that the FRET histogram shifted from this value to this value. We made this distance between donor and acceptor much shorter. And lastly, we add now a full complement, and we have all 4 nucleotides, and now the enzyme goes all the way to the end of DNA and falls off, and we lose all this population. So this is demonstrating how we march the enzyme on the promoter DNA and how we can see distant changes. With this tool, we can now go and answer questions about abortive initiation. Leon, can you help me? I see some more questions on chat.
No, we're fine.
Okay. So we talked about this abortive initiation, the Beetle, Volkswagen Beetle car going up the hill and then shuts off, come down, try again, try again. This is actually an experiment was done in the building I'm sitting in right now at UCLA by Jay Gralla in the early 1980s. He did an experiment called footprinting. Basically, what you do, you use chemical cross-linkers that cross-link the enzyme to DNA. You digest the overhanged DNA with some enzyme that basically chew up the DNA. And the DNA that is protected is then released and analyzed by gel electrophoresis. And you basically look at what is protected by the enzyme. When he did this experiment under the conditions of abortive initiation, meaning he was feeding the system with subset of nucleotides such that it can never go to elongation, you detect all these abortive initiation products. And when you look at the protected DNA on the promoter, you see very sharp end to the trailing edge, but various lengths on the leading edge. That was a huge puzzle that lasted for 30 years. Many papers were written on that puzzle, and it was not understood why the enzyme is doing this. So in contrast to what happened in elongation basically the enzyme sits on the DNA and moves. So it doesn't matter where you are on the DNA. When you cross-link and you analyze, you see that the same signature is protected, the same length of DNA is protected. But in initiation, that's not the case. You get this sharp edge on one end and various lengths on the other. So it's the topic of this paper we published in 2006. And here is the question that we are asking. As I told you, since 1980, when Jay Gralla did his experiments, there was a big question in this field of transcription, how basically we start to transcribe genes. There is a lot of regulation. This is the most highly regulated step in gene expression. When cells integrate all the signals that arrive from the environment, they need to decide which proteins are going to be made at what point in time. And the highest regulation of that process happens on this level of transcription initiation. So to explain J. Gralla experiments, people proposed 3 different models over the years. The first one is inch warming. In contrast to elongation, where the enzyme moves as 1 unit over DNA, in initiation, it's still bound to the promoter, and the leading edge of the enzyme is stretching forward. The bubble is shifting, a small transcript is made, lost, everything snaps back, and you do it again and again. That's an inch warm model. Another model says there are no conformational changes in the protein. The conformational changes are actually in the DNA. And the enzyme, like in a tug war, pulls the DNA in, makes a transcript, loses it, and do it again and again. So one model, the inch warming, predicts structural changes in the protein. The other model predicts structural changes in DNA. The third model is really a more concept about ensemble versus single and time averaging and ensemble averaging. This model is called transient excursion, and it doesn't require any conformational changes, not in the protein and not in the DNA. The enzyme moves in initiation the same way it moves in elongation, but it does it very fast, and then it goes back and stays back for a long time. So the moving forward is very brief, very short. The staying back is long, and when you do those footprinting experiments, on average, you'll see a sharp edge for the footprint here and smeared edge here. So these are the 3 models that were proposed over a period of 15 years before we addressed, 25 years before we addressed this question. And now I've shown you that with single molecule FRET, we can actually measure translocation of the enzyme on DNA. So we can put the donor on the leading edge of the enzyme, an acceptor on the downstream DNA. Note that all 3 models predict that this distance is going to get shorter. So that's a good control experiment, because if we can see that shortening of distance, we know that the experiment is working, and then we can look into other distances. So this is what we call the leading edge FRET, and if we see a shortening of distance, we know that the experiment is working. Then we can do the opposite. We can put donor on the trailing edge of the enzyme and acceptor on upstream DNA. And here, only one model predicts distance changes. This is a transient excursion, other 2 don't. And lastly, we can put the dyes on DNA before and after the bubble and see if there are conformational changes in DNA. And by doing all those 3 experiments, we can come up with a conclusion which of those 3 models is the correct one. So here is the control experiment. We basically prepare open complexes. We add subset of nucleotides. This is the FRET histogram of the open complex. We add nucleotides, and you can see that the center of this cloud is shifting to the right, seeing here in the dashed blue line. And the answer is yes, we can see this translocation. Then we test for transient excursion. I said that only one of the 3 models predict that there are changes in distance on the back end. So we put the labels in the right place. We start with this FRET histogram here. We add nucleotides and absolutely no change. So already we can rule out one model out of the 3. We are left with 2 models. And to tell apart these 2 remaining models, we label the DNA, and we look if we see changes before the bubble, if the DNA is crunched into the enzyme. So what you see here initially, we see 2 populations. So we see doubly labeled DNA, free DNA, where there is no protein at all. So that's a peak that we are not interested in. And this higher FRET peak is when the enzyme is bound and the bubble is opened. And so we are going to look at changes compared to this cloud here. And we add subset of nucleotides, and you see that this cloud that was here is shifting to the right, clearly showing that the DNA is getting shorter. So we basically proved that the scrunching is the correct model. We did it for many different distances, And we were able to show that this model is a correct model for abortive initiation. What it really means is that, what is interesting is, why does the enzyme do this? It seems to be such a waste of energy to try to transcribe, lose a transcript, try again. So first of all, what is the reason that the enzyme runs into difficulty? It turns out that when the transcript that is growing in the active site of that enzyme gets longer and longer, and it gets to position 7, you have a 7 bases long messenger RNA, it hits a wall. Basically, the exit channel of that enzyme has a part of sigma that blocks that channel, and there is a strain built in the system, and that's strain enough basically for the enzyme to abort. The transcript is lost, and it's trying it again and again. Only if it somehow manages to open that gate in the exit channel, it can move forward. And the question is, why does the enzyme do this? So the thought is that this block allows for more time to regulate more precisely the initiation, because once you're committed and you have the messenger RNA, the protein will be made. So you have no longer good regulation, and therefore when you have these strong promoters, you want to be absolutely sure you build this delay mechanism that allows you to interfere. So basically, as I said, when you have long enough RNA, it pushes part of sigma in order to open that gate. So that was an hypothesis in 2006, but then many years later, we came back to this. This is now 2016, and we actually were able to show this very clearly. So what we do in this experiment, we prepare the enzyme at position 0, and we add all 4 nucleotides, and we run the enzyme off the DNA, and we look at the production of RNA. We actually detect RNA here by hybridizing as a donor acceptor single-strand DNA to the messenger RNA that is being produced. And we are measuring how much of messenger RNA is produced as a function of time. So when we do the experiment where we start at position 0, we get a very fast reaction. However, if we prepare the complex, we step it with a subset of nucleotides to position 7. And now at position 7, we add the full set of all 4 nucleotides. We get a much slower reaction. That's a puzzle, but that hints to this blockade that I was proposing before. And if we made this very slow reaction at position 7, but now add another protein called GRIA. GRIA is a protein known to go inside the tunnel and chip off backtracked RNA in elongation. In elongation, there are some pauses, known sequences where the enzyme pauses. And in order to release the enzyme from this pausing, GRIA comes, cuts a small snippet of the RNA, allows to the enzyme to resume the reaction. If we do this now in initiation, instead of having this slow reaction, we go back to the fast reaction. So this actually proves that indeed we had what we call a backtracking and pausing in initiation. This is really coming back to this position 7 that I mentioned before. When the RNA gets to position 7 and it tries to push the gate, it oftentimes doesn't succeed in pushing the gate. And what will happen then, the enzyme will step back, and the RNA will have a tail, which is now backtracked, and the enzyme gets stuck. You really need to add this protein that chips off the released RNA in order to continue the reaction. So this really confirms this concept, and that modifies this bacterial cycle that I mentioned before. You now can add a more complicated steps that are shown here, this pausing and backtracking in initiation that are most likely used for regular gene expression. Any questions about transcription? So I'm going to post today basically a test that will ask you a question about the 2006 science paper. We want to see how well can you understand a paper not in your field. So there will be quite questions about that paper. You will write your answers and submit it as PDF to Moodle. Any questions about that assignment?
No.
Okay. All right, so just to finish off, this field of single molecule threat as a method to look at dynamic structure is live and kicking. In 2018, we published a review summarizing 2 decades of single molecule threat experiments. If you're interested in the topic, this is actually an interesting read. And just last year, a bunch of us in the community published a position paper that really talks about the challenges, the perspective toward the future, and a request for everybody to use open science practices for single molecule threat experiments. So if you open the literature, you can find out that there are many such papers that really talk about what will be possible in the future. This is my former student, Takchip Ha, who also wrote a paper on this topic. And the method as a whole has very wide adoption. Here you see citation from Web of Science. In purple, you see the number of publications as fractions of year. And in blue, you see basically the number of citations. So there are more than around 4,000 publications that actually report on single molecule threat experiments, that are about 65,000 citing articles, and more than 116,000 times basically the word single molecule threat shows up in some papers. So it's widely adopted. And the future for this method, I think, is quite bright. Nowadays, there are some commercial microscopes. You used to have to build the microscope yourself, write codes to acquire data, write code for analyzing data. We used to kind of make this all available and publicly available, but you know, one group doing this was a big effort in the early days. Nowadays, there is an open source that tells you how to build a cheap microscope like this. And there are commercial systems. There is an expensive microscope from Picoquant, and there are cheaper microscope from ONI and EI, and that's becoming much more affordable and democratizing this technique. In this 2018 paper, we talk about things that will be possible in the future, like high throughput single molecule analysis, single molecule threat in live cells, kinetics, non-equilibrium reactions, and so on. This is, again, a paper from the Ha lab showing more than 2 colors. So you obviously can cascade this Jablonski diagram and have threat from green to yellow to purple to red. And now you're measuring actually 3 distances simultaneously. And you can even do it with 4 dyes. So that's getting much more information. Another very interesting development, this is original work coming from the lab of W.E. Morner from Stanford. Morner is one of the laureates for super resolution imaging, where they can immobile molecule in solution. They don't need to immobilize them on the surface. They are in solution, but they are using electrokinetic trap to bring the molecule back to the center when it's trying to diffuse out. And with this, people are now able to do single molecule threat with very long trajectories without the need to immobilize molecules on this, because that always brings some kind of artifacts. The molecules don't function as well as they are functioning when they are freely diffusing in solution. So this electrokinetic trap is really very interesting, and the quality of the data here is fantastic. So I'm sure that in the future, we'll hear much more about this able threat technology. I've shown you the work of Bill Eaton and Ben Schuler about revisiting Stryer and Hageland as a molecular ruler. And Ben continued to work on this, and he's interested in unstructured protein IDPs and looking at fluctuations on much faster timescale that I presented here today. And he has developed these methods to do what is called anti-bunching spectroscopy and fluorescence correlation spectroscopy on threat species. And nowadays using something called zero mode waveguides. These are basically small holes in a metal coated glass cover slip. You get very strong enhancement of the laser in this small hole here. So molecules that come in and out of this small hole will have emitted many more photons, and therefore the bursts will be much stronger, and you can extract with much fewer, a much shorter timescale, you can extract threat values. So he's doing this type of threat histograms and lifetime measurements and anti-bunching to look at these fluctuations of unstructured proteins. And this technology, I'm sure, will be very useful in the future. Eitan Lerner, who used to be my postdoc, is now professor at Hebrew U in Jerusalem, came up with really a very powerful analysis method that can extract different states and transition rates between states for solution-based measurements. This is something that used to be done only on surfaces where you had trajectories, and you could use hidden Markov analysis codes to extract transition rates. It used to be thought that you could not do this for molecule diffusing in solution, and here, again, with advanced analysis, this is actually possible. So this is also very exciting. So in general, I think this is still a very alive and kicking field, and many more new things will be possible in the future. For any of you who are interested in single molecule threat, there is now a community, there is a website, you register, there is a lot of activities that we do as a community. So this is basically the link for this threat community if you're interested. And this is what I had to say today. So any questions? So many of you are physicists, and this is kind of Greek to you, but I feel that, you know, this is important enough, and it really opened up a new field of dynamic structure, and that has significance because you think about drug discovery. All of drug discovery was done until now on static structures that were solved, and people looked for some binders that bind and block the active site. With this concept, think about somebody riding a bike and somebody standing on the ground with a stick and pushing the stick into one of the wheels. The bike rider will immediately stall and fall off the bike. You can think about developing drugs that stop motions in enzymes. You have a hinge motion and you put a peg somewhere and that motion is not available anymore. That opens up phase space for drug discovery in a big way. So I think that this concept of measuring changes in structure as function of time is very, very important in many ways. Questions?
Professor Wייס?
Yes. Shimon?
Shimon, about what you said about the drugs development. If I got you right, and maybe I don't, but if I understand it right, this is just a measurement method. It's not a new function that was discovered. The function was there, but now you can measure it.
Right. I mean, in the past, it was very hard to look at moving parts as general. And I'm saying now that you can use it to build assays that blocks activity in a different mode compared to what was done in the past.
So I won't just measure the end outcome or output, but the process with this method.
Well,
That's
so
what you say?
in drug discovery, oftentimes people use computational methods to dock small molecules into some pockets. And they don't think to dock molecules in other areas of the protein at all. And I'm saying that if I have an assay where I have a moving part and I manage to screen a library and that motion is stopped, I am now docking a small molecule in a totally different area of that protein. And that could really stop. So oftentimes people do drug discovery just looking for the very end result. But this way you lose a lot, you miss a lot. And I'm arguing that it would be interesting to try to develop open phase space and really search for small molecules that block motions on enzymes.
Can I email you an idea that I have developed and we can think together how?
Of course. More questions? Leon?
I would like to add that maybe if you can think about, usually when you think about proteins or like the textbooks images are static. And that's what we get from X-ray, but it's not. So one approach to study protein structure is FRET, and that's what Elisha Haas did for years. He took an acceptor and donors, and he measured those fluctuation in the protein structure. Yeah, and maybe you can think about.
Well, when I started my journey in biophysics, people looked at enzymes as rock solid particles, with a fixed structure, because everybody was used to look at PDB structures of atomically solved structures, and nobody was talking about what are the moving parts and what is importance. And oftentimes enzymes just fluctuate, and this fluctuation is very important. So having tools to look at this, I think, is very important. And I dub this as a whole new subfield of dynamic structural biology. Any other questions?
Hi. First of all, some students from chemistry department have a conference right now, and they ask if it is possible to update the today recording before Sunday.
What I do with the recording, I send it to Rita, and Rita is putting it on YouTube. So the rate limiting step is Rita, not myself.
Thanks. You mentioned that when you got to Bell Labs, you were interested in near-field microscopy, so you build a microscope. So how you approach this task of building something that it is new, new area?
It's a very good question, and let me actually share something here. But this is a question that I can take forever to answer. So let me give you the short answer. So I just gave yesterday a talk about the history of single molecule FRET and my personal recollection. Let's do it very, very quickly. So I mentioned Aaron Lewis. This is his first paper in 1984. He invented the E-beam lithography. That was people he worked with, Isaacson and Murray. And they had an aperture and they wanted to push light through a very small aperture. Then they moved to glass micropipette, not to an optical fiber. That was Eric Betzig's later addition. And I moved to Bell Labs. I know about near field. Then I moved to Berkeley and I want to build a microscope. And basically in LBL, I meet these two guys, Miguel Salmeron and Frank Ogletree, who were already building STMs and AFMs. So they had an experience with building scanners, with piezo scanners. There was no commercial machines at that time. The AFM was just invented a few years before that, the STM 10 years before that. But these guys knew how to build AFMs and STMs. So we wanted to see excitons in quantum worlds, and we started to talk to them. In LBL, we had a fantastic machine shop. So we could design anything, go to the machine shop, and they would make parts for us. So the first student I had was David Botkin, and I asked him to build this instrument. This is a scanning tunneling microscope that was operated by femtosecond lasers. We used photoconductive switching to activate a phenomena on surface and then time gate the tunneling current. And the second student was Tektripa, and I asked him to build this instrument, and this is the first instrument that we used to measure single molecule FET. This is a near-field microscope. And this is a device that went into the evaporator to coat fibers with aluminum metal, such that we can get these small apertures. So, you know, we design stuff and we build stuff. This is how you used to do work in that time. And I think that's very, very important. And that type of skill that you build your own instrument is kind of going away nowadays. Did I answer your question?
Yes, thank you very much.
Any other questions?
Shimon, I have a question.
Yes.
I just want to ask you, like nowadays, like computational biology is like taking more and more into structural biology. Let's say now there is a software called AlphaFold has been coming, and it's like says it's 90% accurate. So the labs like crystallography and microscopy, they are like getting, you know, like less like wet lab work. So
well, David Baker, who is kind of a top guy in this field, whenever he makes prediction, he also makes a protein and crystallizes a protein or cryo-EM the protein, and he shows the computation and the structure as well. I don't think this is going away. Now, cryo-EM has become so good that you can really imagine to have high throughput. You know, it used to be years to solve one structure. That has become, you know, the throughput is much, much faster. And yes, computation gives you a very, very good guess to start with, but still you want to go down to the very details, and computation is not there yet. In fact, we have a very interesting project going along these lines. I talked a lot about initiation of transcription. We want to have a three-dimensional atomic movie of the enzyme on DNA going through all those steps. So we are building microfluidic device. We are doing non-equilibrium reactions. We are making a library of many different dye positions such that we can measure many, many distances as function of time. And then at each time point, we'll have distance constraints, so we can go to AlphaFold and solve the whole complex for each time point. And now we'll have a three-dimensional atomic level resolution movies. So that's, I think, where it's going.
Okay.
More? Okay, so let me summarize. I'm going to post, obviously, the video on YouTube with the help of Rita. I'm going to put the slides on Moodle as usual. And I'm going to put a test that will ask you a question about the 2006 science paper about abortive initiation in transcription. I know that many of you are from very, very different fields. Make the effort to read the paper, to understand the paper. If you have some gaps, try to look for other help. You can just open Wikipedia and read about transcription, or try to answer the best you can. I will not be upset if you answer some questions wrongly. You're not going to lose points. What I want to see is a real, true effort trying to understand the paper. So please answer the questions and post them on Moodle by Sunday. Now, Leon, I'm not sure about the mechanism because I'm not that familiar with Moodle. We are using something else here. But you need to set up an assignment, right, to do that? Such that you drop the work in that one box or whatever. So if
I will
do that,
help you.
Okay. Anything else? well, it's getting late, so good night, everybody. And we'll see you next week. I will be in the East Coast, and I hope that everything will go smoothly. So see you next week. Good night.
Yeah.
Thank you, Shimon.
Good night.
Good night.